I hear it.
Thank you, Shanti. That's brilliant. Okay, right. With the starters gone off, it's my real pleasure to introduce Stefan as is today's IBH seminar. Stefan's been working in the field of meiosis and recombination for a wee while now. He's had his PhD about 10 years ago under working with Andreas Huben, known to some of us, and then a postdoc in Birmingham, where I first met him. And having done his time in Birmingham, he's moved back to Germany, and he's been in the IPK since about 2016. So title of the talk is Towards Harnessing Meiosis in Barley. And Stefan, the floor is yours.
Thank you very much, Luke. I will try. Just give me feedback. I'm more used to Zoom. Do you see my screen now? Luke? Luke, can you see?
Yes, we see your screen.
Perfect. Sorry.
Yes.
Okay, good. Yeah, first of all, thank you very much for the introduction and to give me opportunity to talk a little bit about our research. So as Luke already said, I'm since around 2016 back to the IPK, and I'm basically a cytogeneticist. So I like chromosomes and I like meiosis in the group. And today I would like to show you a little bit what we are doing in order towards potentially harnessing meiosis in barley. So first of all, meiosis is a key event for sexual reproduction. So what is the key of meiosis is actually twofold, because we have 2 rounds of chromosome segregation that follow a single round of DNA replication. And the result of this is on one hand chromosome number reduction. So this is required to maintain somatic diploidy after fusion of male and female gametes. And on the other hand, it also assures a genetic variation. So this is actually by a 2 mechanism. It's on the one hand independent assortment of homologous chromosomes. So if you think about a pair of homologous chromosomes, they can randomly either go to one pole or the other. And the other part is homologous recombination that basically generates crossover, so reciprocal exchange between chromosomes. And all this genetic variation is basically what you require to potentially select for new allelic combinations in the course of a breeding program. So if you check it from a cytogenetic perspective, you can use, for instance, a DNA stain like depicted here, and then you can basically follow chromosome behavior during meiosis 1 and meiosis 2. This process is basically very conserved across the kingdom of life. Here, even if you compare, for instance, barley or Arabidopsis, despite large difference in genome size and different chromosome numbers, basically all starts after DNA replication, that chromosome condense in a kind of a form of spaghetti, so they come closely to each other. And then they can exchange between mom and dad chromosomes, and this results, in the case of barley, to 7 pairs of bivalents, so chromosomes that are physically linked by so-called crossover. And then we have the first and the second meiotic chromosome segregation. Interesting to note is that this process is highly conserved, and specifically during prophase 1, which is very prolonged compared to a normal mitotic cell cycle. It comes to all the aspects that are required to assure genetic variation, so in the name crossover formation. So a common phenomena in plants, so both if you check Arabidopsis, but also if you check barley, that you have a high number of double strand breaks, but you end up with a relatively low number of crossover. So a large number of these double strand breaks is not repaired as a crossover. And this is actually interesting because usually a DNA double strand break is very toxic to the cell and needs to be immediately repaired, but plants somehow generate more double strand breaks than actual crossover. And this crossover or this regulation of which meiotic DSB is selected or DNA double strand break and which becomes a crossover is a very tight regulation. So across the kingdom of life, you can basically find out a few phenomena which I would like to depict here. So if you think a schematic chromosome depicted here, you have initially a high number of double-strand breaks, and from this number of double-strand breaks, only a few really become a crossover. And here we can observe a few phenomena, which we call on the one hand crossover homeostasis. So basically, if you think about a chromosome or a species, you have consistent levels of crossovers more or less, even though you have large fluctuation in DSB numbers. So there's a buffer of crossover homeostasis. And another phenomena is you could think this is on a larger chromosome more pronounced. It basically doesn't matter much, the chromosome size. We always have a phenomena of crossover assurance. So also if you have small chromosomes or large chromosomes, typically we find at least one crossover. And most of the time we find 1 to 3 crossover per chromosomes. This one crossover is very key to keep the chromosomes together and assure proper segregation during divisions. Another interesting phenomena also conserved across the kingdom of life is a phenomena of crossover interference, which has been long known, so which also creates genetic linkage. So if you have, let's say, 2 crossover per chromosome, they are usually much further apart from each other than you would expect by chance. So this results that typically crossovers are much more widely spaced and you have very limited crossover form to each other. So which you can depict here. So if you have 2 crossover, normally they are far more spaced than you would randomly place them. And importantly to say, in plants, including barley, we have this so-called interfering crossover that we call class 1 crossover that depend on so-called ZMM proteins, and these are around 85 to 90% of the crossover. But you can also infrequently observe, like depicted here, other type of crossover arising, which we call the minor crossover or class 2 crossover that do not depend on the ZMM proteins and that are also non-interfering, so they are more randomly placed. And these are around 10 to 15% of the total crossovers. All this decision, which DSB or what becomes a crossover, does not take place somewhere. It actually takes place in the form of chromosomes or the chromatin. So during meiosis, we have very specific structures formed in the cell. So the sister chromatids are itself anchored in a loop-based array along a proteinaceous structure, which we call the meiotic chromosome axis. So which is depicted here, mum chromosome and pap chromosome. And initially, double-strand breaks are induced in this proteinaceous axis. In this proteinaceous axis, we know many of these components, like depicted here, forming like spaghetti-like structure in the nucleus. And then some of these DSBs, if they are repaired on the homologous chromosomes, brings basically homologous chromosomes more closely in connection, and then start certain proteins to assemble, so-called central region proteins or zipper-like proteins like ZIP1, which you can also see here. And then they gradually spread from this recombination site, physically linking the homologous chromosomes at a distance of around 200 nanometer. And interestingly, very recently, we also have learned about more proteins that are sitting in between these chromosomes. You could ask, why do I tell you all of this? Actually, it is very important that they really dictate crossover patterning. So if you have mutation in this kind of genes, you can have different, in the case of a number of crossovers or differences in patterning of crossover. And all this tight regulation results typically, if we stain here, for instance, in Hordeum bulbosum, we use a basic stain and we see the metaphase 1 chromosomes that we see for the most part only ring-like structures or like rod-like structures. So this means we have either 2 terminal crossover or only 1 terminal crossover. And this in turn also means that large regions or interstitial region in these chromosomes basically never recombine. And if you think this more on a chromosome or molecular level, if you see here, for instance, barley chromosome 3H, and you plot the gene content versus recombination rate, you can see that basically recombination rates occur at the end of the chromosome, which is very similar to the distribution also, for instance, in wheat, where we see this strong U shape. You can also plot this normalizing across from telomere to centromere in different species. So for instance, here barley and wheat, or comparing it to Arabidopsis. In both plant species or both plant types, you can basically see that centromeres are devoid of a crossover recombination. However, compared to cereals like barley and wheat, where recombination rapidly drops down after the telomeres, in Arabidopsis, actually you have along the chromosomes quite high numbers of recombination. So it's very interesting also to use these two species in order to understand what are the difference or what are common mechanism. So these are basically the two models we also use in our research. Before I start, I don't want to bore you, but I would like to have a short discourse, maybe 2 or 3 minutes on Arabidopsis research, how we use Arabidopsis actually to identify new meiotic candidates that we actually immediately also translate into barley. So one way to identify chromosomes is proteomics based. So here we use the so-called turbo ID approach. So what is turbo ID approach? So my colleague Chao Feng was so keen to explore proximity-based labeling meiotic cell. So he was using a biotin ligase. He was key now already quite some year ago, the description of a very potent biotin ligase called turbo ID. So if you fuse this biotin ligase to a protein of interest, you biotinylate protein in the surrounding, you can capture them, and then you can perform MS analysis. So Xiao was too keen to explore whether you can do in a very limited number of cells, so these few meiotic cells that you have per plants, whether you could perform proteomics. So the idea was very simple, using two known access proteins to biotinylate the meiotic chromosomes and try to identify proteins that are sitting there at this time. So this Chao actually did successfully. So he used two known meiotic access proteins, and he could show that upon presence of turbo ID, we can beautifully biotinylate meiotic chromatin, whereas without the turbo ID, you do not see any biotinylation. Then together with colleagues in Vienna and a lot of mass spectrometry, we could find a number of proteins that we termed access turbo candidates. So either specific or shared between these two access protein. Most importantly, we could identify basically all known access in the C protein in plants, but we also identified a number of new proteins. And one of these new proteins, we characterized further, and I would like to show you that the candidate number 21, we recall to SCEP3 for synaptonymic complex element protein 3. So that is a novel SC-related protein, so synaptonymic complex that physically connects homologous chromosomes during meiosis, enabling crossover exchange. So using here antibodies against Rec8, so which labels the two homologous chromosome axes, using an antibody against ZIP1 in magenta, which labels the zipping between the homologous chromosomes, and antibodies against our protein. I hope you can appreciate that our protein localizes to the center of the SC, so being a C protein. Now you could ask, why do I tell you about a novel SC protein? So we did reciprocal backcross population to map recombination frequencies in offspring, distinguishing male and females. And whereas in a wild type scenario, you see a phenomena of heterochiasmy, so you see difference between male and female crossover rate. This difference between male and female crossover rate is abolished in the mutant, and moreover, you see even an increase in recombination. So this is depicted here. So you have now both in the male and the female similar recombination rates, and you have also an increase. And most strikingly, when you check in male or female in a wild type scenario, and you check or focus on chromosomes that have exactly two crossovers per chromosomes, you can see if you measure the distance between these crossovers, this is far more larger than you would expect it by chance. So if it would follow a random distribution. So this is again reflecting crossover interference, that crossovers are more widely spaced than by chance. And if we check this now in our mutant here, you can basically see that also crossover interference is abolished. So depleting this protein, at least in Arabidopsis, results in increased recombination, abolishment of male and female differences, and you do not see a phenomena of crossover interference. So this was one way to identify new meiotic candidates. When we started, many years ago as a meiosis or cytogenetic person to study really meiotic recombination in barley, we were first developing certain tools which I would like to introduce you before in a later talk, showing you various meiotic mutants and how they impact meiotic recombination in barley. So first of all, if you want to assess meiotic recombination outcome, one typical approach would be to perform, for instance, genotyping by sequencing or molecular marker analysis. So in this case, you depend on segregating population. An alternative from a cytogenetics perspective could be meiotic chromosome analysis. So you could either use a DNA stain, so quantifying bivalent or chromosome shape. So you could very conservative calculate 2 or 1 crossover, or you could use antibodies that label really recombination or crossover site like depicted here, and you could quantify this. But here you're also depending basically on a low throughput, and this is also highly laborious. So in the beginning, when starting this work, we asked whether we could omit segregating population in cytology and instead use gametes, so the direct outcome of meiosis. So whether we could use pollen nuclei to really measure fast and rapidly meiotic recombination outcome. That in principle allows you that you have from single individual hybrid plants, you have large amounts of pollen. Even from single plants, you could immediately measure crossover frequency in this pollen, and you would have no need to grow large segregating offspring population. So this allows you really from a research perspective or also from an applied part to check how the influence directly on a plant is on a recombination rate. So this is depicted also here. So this is a barley pollen. You see a rigid pollen wall, and inside you see the sperm and the vegetative nuclei, so which are the haploid outcome of meiosis. And typically we speak here around 5 picograms of DNA that is haploid. So initially such tools were developed, for instance, also in the group of Andreas Huben, that we were able to isolate pollen nuclei, but due to the limited DNA amount, it was typically required to perform whole genome amplification to later on sequence or genotype a low number. We decided for a different approach, so rather focusing on a few certain chromosomal regions or intervals in barley, and rather to increase the throughput. So already some years ago, we described this method. So it's basically depending on isolating pollen nuclei. You use a flow cytometer to use this DNA stained pollen nuclei to sort according to the ploidy. So you sort haploid pollen nuclei, and then we embark on droplet digital PCR or crystal digital PCR to, in a small reaction volume, basically genotype a high number of individual pollen nuclei. So this was here pioneered from Jun Jae, very talented PhD student, and Jörg Fuchs, who is in charge of the flow cytometer. And how this looks like, you can see here. So if you zoom in, you see 10,000s of individual droplet forms. And then due to the stain of the DNA of this pollen nucleus, you can beautifully see here, for instance, depicted in an individual pollen nucleus encapsulated. And then here, the color basically implies that you have a genotyping reaction going on. So in this setup, we can basically analyze 12 samples in parallel per run in such a digital PCR, and per run we can generate around 25,000 droplets. So first of all, we were also interested whether what we measure in pollen more or less reflects reality. So we were comparing recombination rates in pollen versus the offspring of these plants. And when we did this, basically what we saw is that there is not really any difference between recombination found within these intervals in offspring plants versus pollen recombination rates. More recently, we have continued to work on this. On the one hand, for us, it was a very limiting factor that we were relying on fresh pollen. So we were not able to also use, also from collaboration partners or from different experiments, to use also frozen or stored pollen. So we basically isolate anthers and snap freeze them at -80, and then we can later on process them. We also increased not only the throughput, so now we can analyze with a new updated machine up to 48 samples, generating each time 18,000 droplets. We can now also measure from a meiotic perspective or applied perspective, importantly also crossover interference. So now we have different genetic intervals that are genetically linked, and accordingly we can on a single haploid pollen nucleus also check within flanking intervals whether we have the phenomena of interference or not. So this is basically work of two PhD students, Surya and Michael, who are both doing really great work. So what you can do with it, just as a snapshot, so you can now analyze also different impacts, like it has been shown also from the colleagues in Dundee, like temperature, but we asked various different things. So we had in mind, because it's well known from Arabidopsis or other species, that during the age or the development, the recombination in sperm, for instance, in human, differs. So we asked whether a similar situation could be also found in barley. So we basically took anthers. These are from which we call it later off-center, so apical or basal anthers, or we isolated pollen from intermediate sections of spike, and we also sampled pollen from this, from so-called primary, secondary, or tertiary spike to see whether there's with age or depending on the position within the spike, we can find difference in recombination rate. But to our surprise, it seems in a single barley plant, actually meiotic recombination seems to be tightly regulated. So we don't see really any dramatic variation or tendency that apical, intermediate, or different age spikes show difference in male recombination rates. Another bottleneck is to generate mutants in barley. So for this, usually you depend on genetic transformation and plant regeneration. So together with Jochen Kumlin, we asked already quite some years ago, whether we could skip this. So whether we could, for instance, generate plants that are constitutively express the Cas9 protein, but no single guide RNA, so not addressing any single gene. And then we asked whether we could use an RNA virus, so the barley stripe mosaic virus, load this BSMV virus with this guide RNA and with the scaffold for Cas9, and then infect this Cas9 expressing plant. And the idea was very simple. We have an RNA virus, we have the Cas9 protein. Can the guide RNA within the RNA virus and the Cas protein form an active Cas9 complex and generate editing in an affected plant? And this is here depicted one of the first experiments which we did. So we targeted CMF7, so an albino or variegated phenotype, or a gene that is known to cause this phenotype. And when we sampled from individual infected BSMV plants, we could see actually, if we sample all across the barley plant, high frequencies of editing events. And then when we took randomly from different individual plants the offspring, we could already note directly in the next generation that we can different types of albinos recover, so that we have actually heritable editing at this locus. Then we asked another question whether the virus, if it's seed transmissible, and this is actually the case. However, in the offspring, independent of the presence or absence of the virus, we can basically find heritable edits. So with this, we are now able to induce heritable mutations and to isolate various mutants. So with these tools in hands, we have studied various different meiotic genes in barley with the aim to modify meiotic recombination outcome. And now in the last minutes, I would like to introduce some of these meiotic mutants. Just a short recap. We have a high number of double-strand breaks, but a very limited number of crossover. Most crossovers are class 1 crossover, a limited number is class 2, but most are non-crossovers. So over the last many years, now majorly in Arabidopsis, but also now increasingly in other plants, different factors have identified that can change this equilibrium. One is, for instance, so-called pro-crossover factors. So if you overexpress these, for instance, you can basically get an increase in class 1 crossover. Otherwise, we have various different genes that are so-called anti-crossover factors that disassemble certain recombination intermediates and channels them toward non-crossover. So the result, if you knock them out, in most cases, you shift this equilibrium, you get more crossover. And last but not least, as I also showed you in the SCEP3 mutant in the beginning, but also more recently in other mutants of the synaptonymic complex, it has been shown that the SC, surprisingly, it stacks chromosomes together. But at least in Arabidopsis, if you deplete this SC, you get an increase in both classes. So we asked basically what happens in barley if you generate such kind of mutants. Initially, from a research perspective, we generated also a mutant in a gene called mTOP6 beta. So mTOP6 beta is a gene that forms an active, let's say, complex to induce double-strand breaks. So when we generated mutants in barley, we could see that there is no difference in the growth of the barley plants, but these plants are completely sterile. So if we check now the meiotic behavior comparing wild type and this mutant, whereas in a wild type we can see crossover recombined chromosomes, in these mutants we basically see complete absence of crossover formation. So knocking out this gene, you can basically shut down completely homologous recombination in barley. Now I would like to introduce you to a few pro and anti-crossover factors. So here I would highlight specifically Mo Ayub and also Maria Kouakos, but also many other people. So high-10 is known to be a sumo or potentially even ubiquitin E3 ligase, and we asked whether high-10 is also a pro-crossover factor in barley. First of all, we generated mutants to different mutant alleles. We generated specific antibodies showing that in both mutants allele we have complete absence of this protein. And moreover, most importantly, using this antibody, you can see here these red dots, which are basically reflecting the crossover site in barley. So you can beautifully cytologically quantify and also place along the chromosome the crossover sites. So how do plants look like? High-10 plants are basically looking like a wild type. However, they have a very strong, or they are nearly sterile, these plants. If you check the meiotic behavior, the sterility is based on limited meiotic recombination. So we see a lot of univalent chromosomes that randomly segregate, leading to aneuploid gametes. And quantifying the residual number of chiasmata, so the minimum crossover number cytologically, we could also clarify that around 90% of the total crossover in barley are so-called class 1 crossover, whereas the residual ones are so-called class 2 crossover that are randomly placed. Moreover, when we quantified in a heterozygous state, so we only have one functional allele of this gene, both the high-10 foci number, but the minimum chiasmata number, we could also see that this gene shows haploinsufficiency. So half of the dosage of high-10 leads to a reduction in crossover. So it's very interesting also to ask the opposite way around. Another gene was fidgetin, which is a so-called anti-crossover factor in Arabidopsis, but in other species such as rice, it has been shown to result in a complete meiotic catastrophe, so complete sterility. We still went ahead and generated different mutants. I would like to focus today on 2 null mutants. If you check the barley growth phenotype, you see in a homozygous state that you have a slightly retarded growth, and you also have a reduction in fertility, but you only have a slight reduction in fertility. Checking chromosome behavior in barley, we could show that we see a low frequency of univalence, so chromosomes without crossover recombination, and that we had a reduced number of chiasmata. This sounds not so, let's say, interesting at the first glance, but we were interested how it is really, and it's an interesting phenomenon that this gene is actually both an anti-crossover factor, but at the same time it's also a pro-crossover factor. So if you quantify class 1 crossover formation, you can actually see a drop in the fidgetin mutant. However, if you introgress the fidgetin allele into a mutant that cannot produce any more class 1 crossover, so the high-10 mutant, you see actually a large increase in bivalent. So you recover again crossover formation, suggesting that this gene limits class 2 crossover. So this gene has actually dual function. At the same time, the retarded plant growth also suggested that it might be important for DNA repair. This we could also see in a few meiotic cells and somatic cells, that we can see chromosome bridges or chromosome fragmentation, and at the same time we performed assays using DNA damaging agent to ask whether these plants are more sensitive than the wild type towards these drugs, and this is actually the case. Whereas the wild type shows some sensitivity, the mutant shows strong or hypersensitivity against the DNA damaging agents. So fidgetin is also needed for genome stability. We also generated a mutant in different cultivars of RECU4. So RECU4 had been also described by the colleagues in Dundee as an anti-crossover factor in barley. We generated in parallel also various mutant alleles. And in the case of RECU4, we see a slight reduction in fertility of these plants. However, if we check meiotic chromosomes carefully, different to a wild-type scenario, we see many chromosomes with such protrusion, suggesting directly that you have additional crossover along the individual chromosomes. We used pollen typing in different genetic intervals, and we could see in average that we have a threefold increase in recombination in hybrid plants. However, in the case of RECU4, we also can note that we see certain genome instability, so we can see fragmentation both in doing mitotic and meiotic cells, and we could also see that treating these plants with DNA damaging agents, that we have a hypersensitive response. Last but not least, how is the axis and DSC in barley to basically close the story? So generating antibodies against all these axis and SC components, we could see that DSC in barley is conserved. And this is basically work of Maria Kourakos, Zuria, and Chow. And when we later on analyzed this mutant, we could see that these plants are fertile. And know that the plants have they look vegetatively fine, but they have a strong fertility on grain setting per spike. When we check the meiotic phenotype in all cases, we could see a reduced number of bivalents. So different to Arabidopsis, DSC seems to be required for crossover formation, but it does not limit crossover formation in barley. Most importantly, when we check the remaining chiasmata or crossover, they are randomly distributed, suggesting that residual crossover in this mutant show a random distribution. And with this, I would like to summarize. I showed you today something about single pollen nucleus genotyping and barley stripe mosaic virus-induced genome editing. I showed you something today about mTOP6B that is required for crossover formation, about a gene high10 that regulates dosage-dependent number of class 1 crossover, about fidgetin that limits class 2 while it is required for class 1 crossover, about a gene RECU4 that limits class 2 crossover, and that DSC and the name ZIP1 and SCEP3 that they are required in barley for crossover formation. And with this, I would like to acknowledge the people who are actually doing the work. I would like to thank a lot of people at the IPK for help and constant support, and a lot of people outside of the IPK. The list should be much longer for sharing material, expertise, and nice discussion. And last but not least, the funding bodies and you for your attention. Many thanks. Luke, I can't hear.
But after all these years, I still don't remember to unmute. Yeah, thanks for that. Whistle-stop tour of a really fundamental process in all eukaryotes, but particularly interested in large genome serials. Could I ask anyone either, if they want to ask a question, either to raise their hand or ask a question, or to probably put something in the chat? If I could just kick off. I'll pass over the Arabidopsis work if you don't mind, Stefan. But I was really interested in one of the slides you had where you had the conservation of recombination frequency within individuals. Did you find significant difference between individuals in the same?
I mean, we have here basically asked in the beginning, first of all, for individual. Let's say if there's a differences, you could maybe use different unpairs also for pollination, very similar. Mm-hmm. And then we have also compared different individuals growing, and there we can see variation. I mean, this is also you find across most of the plants. So if you pick individuals, both Arabidopsis or IBH barley, you see some variation in recombination rate. But overall, if you take multiple individuals, what we most of the time do to pool it, we basically get similar between when we take smaller populations.
So have you used that technique to try and tease out individual differences at all? Or, I mean that's a really powerful technique you have there.
No, we haven't done, because I mean, it would be something, but we have decided for all our work to really find something more generally, because you have this plant by plant variation and recombination that we can find, and we are rather going in the approach, pool a few plants to balance out this plant by plant variation and see really a genetic difference or really modifier.
And from that work, you were the last thing you just touched on very briefly, from your work in barley, the SC components you're saying are required, but they don't seem to be determinant on. I mean, unlike Arabidopsis, you're getting a very different mutant phenotype.
Yes.
Do you have any hypotheses, thoughts why that is? I mean, because Arabidopsis is, for better or worse, the model plant used in a lot of studies.
Yes, I mean, there you see also it's an interesting, this SC is highly conserved really across the kingdom of life, but this requirement of the SC for crossover formation also differs. For instance, when you see a yeast, in yeast, it also limits crossovers. It's very similar like Arabidopsis, but you have other species, mammalian species, where it's required for crossover formation, and in barley, this seems to be also the case. It will be very interesting to see whether this has something to do with genome size, that you have maybe, you need, how to say, to recombine still the chromosomes need to be able to find each other. So if you have this large chromosome in a large nuclear space, maybe you have per megabase relatively few double strand breaks. So it's very unlikely that, homologous chromosomes find each other if they do not really get physically connected. Very simplistic view.
Yeah, no, but it's, yes, it is illuminating how the certain phenotypes, the model plant is not necessarily a useful model.
Exactly. And I mean,
Yeah.
I think also barley as a model. So it's very nice to use barley and both as a model. And it's also very nice from a meiosis perspective that, there are really large difference between the species.
Okay, we have a question here from Clara, one of the students here. Clara, do you want to ask the question or shall I read it out for you?
Where is it? Where is it? Chat. Chat.
It's in the chat. Yes, welcome to WebEx, Stefan. I'll read it out. She asks why the lack of recombination causes such a strong sterile phenotype, and a really interesting question as well. Also, how is there a way these techniques could be used for looking at female meiosis, female gametes?
I mean, the strong sterility phenotype is relatively easy to explain. So if you have a pair of chromosomes, if they have a crossover, they are physically connected, so the two cell poles can separate the chromosomes equally. So the higher the chromosome number, if it's not the case, these two homologous chromosomes, so the mom and the pap chromosome, randomly are distributed. So the higher the chromosome number is, the less likely it is to get a gamete with a proper chromosome number. So if you would have a species only with one chromosome, you could get this, despite recombination, basically 50% proper gametes. But by every time it decreases by half. So if you have 7 chromosomes in barley, the likelihood to get really a proper gamete is incredibly low, resulting in these. To analyze female gametes, female gametes is simply incredibly problematic to assess. In theory, it would also be possible, but they are much less abundant than, let's say, male gametes. So they have some ways showing that you can also sequence female gametes. One other common way to maybe check female meiosis is maybe rather to do reciprocal back crosses and then to see how the recombination rate in the female is. But yeah, for sure, male and female, there are differences in recombination. So we check for the most part from a meiotic perspective in males, but females is the other side. Yeah.
Another question that came to mind is that is there a difference, because one of the advantages of the system you're generating, you're looking at potentially generating multiple alleles. Do you find allelic differences between your induced mutants, or are you screen for more stronger phenotypes before you do the strong analyses?
I mean, look, from a research perspective, we normally we try to find null mutant alleles at the moment in barley and to check how it is, because it always takes a certain time and also effort to analyze it. But definitely, if you think about also a different mutant like involved in DNA recombination, like the RECU4, if you could unlink the somatic and the meiotic function, there are potentially approaches like you have seen in model species of such a DNA or modifiers, or also the SC, if you could find potentially, let's say, truncations of the protein. Maybe you could bring the chromosomes close together. Definitely, these are things. But yeah, null mutant are preferred for the most students and for us at the moment.
Next talk, maybe. Sebastian, you have a question?
Yes, thank you for your talk. It was very interesting and that's quite large story. I have, actually, my question is, you mentioned multiple of your mutants were completely fully sterile. So my question is, did you test if different growth temperature, like increasing the temperature, for example, might let your plant pass through the meiosis and have a proper segregation of the chromosome and restore fertility?
Short answer, we haven't done this properly. Longer answer, it's a very interesting thing to proceed, and we are also trying, because you see already in the normal, let's say in our case, the normal greenhouse condition, you see variation between individual plants in this mutant. So there's definitely the environment will have an impact. So it's something which we have on the radar.
If you have a growth chamber, I will suggest to you to test 24 degrees first.
Yeah.
It worked for some other mutants, so it may help.
Yes, thank you.
Stefan, I hesitate to bring it up, but do you have a feeling of any of your mutants actually changing the distribution of recombination as well as the frequency?
I mean, it's interesting, for instance, if you quantify, for instance, even, let's say, in this high 10 mutant, where you have an incredible low number of crossovers, the crossovers which you have there, for instance, here at the very, very chromosome end. And this seems to be also a very common case also in the SC mutants. You have the feeling the crossovers are even more terminal than usually. This could be somehow related to the bouquet, that the chromosomes there find each other first, and this is the best probability to get a crossover. So there's for sure differences in this distribution, but mostly they go even rather more terminal.
Yeah. So any thoughts, is a leading, but any thoughts at all on how you could target these recombination poor regions of the genome?
I think here, I mean, it will be interesting. Let's say we also don't know, even in a model species, really what can, let's say there's some indication. Epigenetic modifications could be quite interesting because there's a correlation. It would be interesting also to try to identify in model species what really limits, let's say, centromeric crossover, because there are indications you have double strand breaks and there are some indications for homologous repair in the centromeres. So what limits it? I think there will be ways in future also thinking about targeting certain recombination proteins. Is an interesting phenomena. It would be also interesting to make chromosomal rearrangement to see how this behaves. So I think there will be ways.
We've got another question in the chat. I don't know if Yung-Ching wants to ask it. If not, I can say she's asking, is there a difference in the recombination rate between inbred and outcrossed species?
I think it depends for the most part on the species too. And I mean, we don't know too much also about outcrossing species. I mean, particularly from a meiosis perspective, I mean, really work on an outcrossing species might be from a research perspective, from a meiosis perspective, very hard. But I think it would be very interesting. So I think also working on rye also would be something very interesting. And now with the pollen typing, it's something to address also in rye. However, yeah. So I don't think, let's say, from model species, there's really a difference.
Sebastian, is that hands left over from previous, or have you got another question?
I mean, I forgot to remove it, but actually I may have another question, so it's not a bad thing. It's a little bit a follow-up question about what Luke asked before. What determined the location of the recombination? Because it doesn't seem to be random. It's more heterochromatic than achromatic rather than heterochromatic. You mentioned about the epigenetic, but are there any kind of DNA sequence that let the machinery to recognize the right location to give the crossing over position? And if so, would it be possible to create a synthetic crossing over location in the genome to kind of break the linkage group? What do you know about it?
Sebastian, so I mean, this could be a very long answer, but basically starting from the beginning, let's say it starts here always a double strand break. We know all across the kingdom of life, let's say that we have for sure double strand break hotspots. So certain region in the genome that more double strand breaks that by chance. That is normally typical, let's say you could call it open chromatin. This very commonly also correlates with gene promoters or gene terminators or UTR. They are typically AT-rich regions, so most species you have AT-containing motifs where it goes down. This comes more to the, you could say, opportunistic nature or of this topoisomerase 6 complex that actually induces the DNA. This has more affinity, accessibility for open chromatin. Accordingly, various epigenetic marks are also, you could say, depleted. And then the procedure continues the same also on the crossover pathway. You can only have the same, let's say, crossover hotspots all across the genome, regions which are preferred to have more or not. There's a correlation towards double strand breaks, but it's not the whole story. So it's not per se where a double strand break is, you get a crossover. Because also if you check in barley using cytology, which is very hard to trying to quantify DNA repair sites in barley, I mean, you see it more or less all across the genome. So there's a selection.
Will it be possible to demethylate the DNA with some chemical and to test how it could interfere or impact with the crossing over rate?
Exactly. I guess these are also people doing or have tried, or there are also mutants available. But I think it's only half of the story. I mean, you get the open chromatin, but let's say in the Arabidopsis and model species, in a few of these mutants, you can get crossovers where you normally don't really have, but there might be more requirements.
Definitely, the other thing you brought up was interference. And looking at, I think for me, some of the intervals you're studying in your pollen, some of those intervals go across the centromere. So potentially the distances over which interference is working in barley are very considerable. I mean, is there any thought on mechanisms or how that is maintained?
I mean, this is at the moment, let's say, you could say one of the hot topics in the meiosis field, particularly in plants, what could be interference. There have been a lot of models proposed at the moment, basically majorly based on plant knowledge. The so-called coarsening model of this protein called HA10 has been proposed, which is kind of, you could say you need the SC is the idea, and you need this protein. So you have like a tube, like a railway, where this protein coarses, like a certain tendency to self-accumulate, and then gradually it gets bigger. And this seems to work over relatively large distances. So we did this in barley using simply also grinding across the centromere to ask whether, let's say, the centromere is a boundary. So in this case, we actually get rather positive interference. So the centromere seems to be, as across all species, you can rather see interference on the individual chromosomal arms. So across the arm, you have interference, but the centromere, it starts to reset, you could say. So you see this also in barley. Typically, you have one crossover on one chromosome end, and you have another crossover on the other chromosome end. And not very often additional crossover along the individual arms. And if so, normally further spaced than usual, you would expect.
I have to think about how that works in terms of mapping. But you still get linkage across the centromere.
Yes.
Yes, but not necessarily the.
So if you would use, let's say, an interval across, let's say, a chromosome arm, and see that you have interference, so that you have a chance in these two flanking intervals to cross over at the same time is lower than you would expect it by chance. However, if you have, let's say, you take an end interval and you take another interval going in the additional chromosome, you basically see that interference diminishes over the distances, and at some point it even disappears, because typically, for the most part, you will have on both arms a crossover.
Mm-hmm. Yes, that makes sense. Yes, but so the mechanism, even in the genome size of barley, the model seems to hold.
I mean, it seems to be, let's say, it is very still enigmatic. So in barley, you could say, argue at the moment that DSC is not necessarily, or let's say, there are different ideas how this interference signal is transmitted. On the one hand, is considered to be DSC, seems to be required.
Mm-hmm.
I10. So if you have more dosage of I10, you get more recombination intermediates. If you lose DSC, you lose interference. However, that seems to be at the moment not the case, at least for DSC in barley. So what is really the interference signal spreading here is something to be, nobody knows. I mean, this is already described for more than 100 years, and still there's a large debate what it is in reality. There have been a lot of models proposed.
Okay. Just double check. Is there any more questions, Stefan? I've kept you long enough. I mean, we have apologies from a number of people who can't be here, including Isabel, obviously. But thank you so much, Stefan, for a very comprehensive tour of barley meiosis in half an hour or so. Just to let people know, the talk eventually will make it onto the internet as well for viewing for the future. So thanks again, Stefan. Thanks for your time, and thank you everyone for patience. And hopefully we'll see you again at the next seminar. Thank you.
Thank you for listening.
That was great, Stefan. Covered a lot of ground very clearly.
Yeah, it's simply I was not really sure also how much to go in detail or what to do, because I think for this seminar, trying to keep it simple also when the answers, now you're asking questions. I mean, you know, you can talk about all the proteins and players, but I guess for the most people,
Yeah. No. It's no, that was
to keep it simple. But if you sometimes keep it simple, it also gets tricky to explain properly.
But no, I think the questions were sensible as well, and you know, they led on to some nice discussion. So, no, thanks.
Yeah.
Yeah, I think the combination maybe between inbred and outbreeding, looking at bulbosum, it seems to be even more terminally distributed. And,
Exactly.
You know,
I mean,
if anything, the inbred has relaxed things a little bit, even though they're still incredibly distally.
I mean, we did this here also with Martin and so on to check this all with bulbosum, and we also did cytologically. We even have a feeling it's even more distalized, the crossovers in bulbosum. There might be differences, but I mean, you would have to study really a lot of outcrossing in breeding species. I don't think it's so easy to say.
I mean, do you feel there would be a signature in, I don't know, SC protein sequence or anything that you could use that once? I mean, I'm thinking of the Hordeum. Now you're getting all these. Is there a way you could start looking at differences across some of those species, some of those different interspecies differences, or is that too messy?
I don't know if it's too messy. I mean, there have been a lot of, let's say talking about meiotic genes, a few genes have been identified all across the kingdom to be, let's say, under selection in terms of ploidy, in terms of higher, lower recombination rate. So it would be also interesting now to have a more look on this. But I mean, I guess you did the same. We did also in barley to check for all these guys if there's a lot of allelic variation, but that seems to be not so much the case.
But potentially your pollen assay, you could use that across multiple crosses, multiple combinations.
Exactly. This was also the idea, but it's still, let's say, you know, to have a proper marker running on the pollen. We rely here on Heckmann probes, so it's always not so easy to have really very good markers.
Mm-hmm.
If you have the markers, let's say it still also takes a lot of time. So the idea is for sure, or was initially, to check it all across for variation recombination rate. If this is the question.
Yeah, no, absolutely.
So, I mean, this is here what we also discussed, let's say with Isabella in a project, I would like to have then also marker for this bulbosome introgression and then track simply, you know, can you increase or lower the recombination? So you can basically get snapshots, I guess, from this introgression lines perspective. It's quite useful.
I mean, we're generating some SNPs at the moment, he says. Yeah, but as soon as we get any information, I can share that with you, because it's, you know, it'd be kind of interesting to see. It's amazing how, yeah, in some respects, how I was surprised even some of our standard 50K platform, some of those SNPs worked fine in an interspecific hybrid. So there's obviously enough conservation.
Yeah.
Obviously, a lot of them were nulls and things, but there was some that seemed to work in a codominant fashion. So, yeah, I think it's not insurmountable.
Yeah.
And I guess now with the sequence information there, that you can target that.
Yeah.
Yeah.
How is Isabel doing, by the way?
She's literally in France. That's funeral, I think, this week. So, yeah, she was obviously in a bit. She had visited over a week or so ago, which is great. She actually saw him before he went, but yeah, I think there was a lot of emotion. So
yeah, no, I felt. I mean, she was also. We had agreed that she would come a little bit, and for sure, she should go to her family and everything. And I saw in the emails that she was, obviously. I hope it will be fine. I don't know.
Yeah, no, I think there's a lot of history and stuff that she's family history that it's not always obvious and not always easy for her, I think, as well. But I think it's been I'm glad she went over before, and it's good that she's reconnecting with her family and stuff back over in France as well. So
look, we should also, I guess, soon, even now from this call or this trilateral call, I assume there should be also here something, even though I think the chances might be very limited, but yeah, we'll see.
Yeah, indeed. You never know. And there's always, it's a basis for something else if not. So
yeah, no, I will be also then coming in February there on this Barley genome that meeting. I said I will come, then maybe we can also see and have a chat or something.
Yeah, absolutely. Yeah. It was good to see you, even on in a WebEx. And yeah,
it was also the first time for me. It's actually super confusing, this WebEx, when I use normally Zoom. I did not see you all. You have no feeling. Even though if you only see the names there listed, it's super weird. You talk very different than the Zoom. They normally see some faces or whatever on top line. It's completely weird.
Yes. No, you did well. Often WebEx can go horribly wrong, so it all went well. Thank you.
Good.
Okay. Good. Cheers, Stefan.
See you. Bye.
Bye.